Morning.
Morning.
We've given you permission to share your screen.
Excellent, thank you.
All right.
Okay, I hope you can see my slides. As I've mentioned, we're going to go through Salmonella, Shigella, and Pseudomonas. And in case of any questions, you can put that in the chat, or we can discuss them as we go along. So again, I've mentioned that these pathogens, particularly the first two, Salmonella and Shigella, continues with the lecture series that I believe you've already been introduced to, bacterial diarrhea or stomach pathogens that cause either diarrhea or stomach upsets or gastrointestinal complications, as this slide really depicts, where we're going to look at Salmonella, we're going to look at Shigella, and study the two pathogens in more detail and to see how well we can prevent it. So really our objectives today, we're going to look at these two pathogens first, look at the general characteristics, the different virulence factors. Remember I mentioned last time we met the importance of virulence factors. These are factors that make this bacteria fit enough to be able to colonize the host, cause the infection, and thrive inside the host, despite the host mounting a very effective immune response. So we're going to look at that, and then we're going to look at the pathogenesis of these two pathogens and see how they cause diarrhea, and also see how to differentiate between the two pathogens depending on the diarrhea that it causes, which is very important for us who'll be in the lab trying to make the diagnosis. We're also going to look at, of course, I've mentioned the lab diagnosis, but also look at treatment and how to prevent. You'll see I've asked you which of the two has a vaccine. Hopefully you can be thinking of that. And then we're going to look at, touch on Pseudomonas. Most of us in our clinical setting, we normally call it pseudo. You see why, again, is an important pathogen to be able to understand and see how we can prevent it. So I believe most of these pathogens have been introduced to you, and if not, they're going to be introduced slowly as we continue this lecture series. I believe we've already looked at E. coli and several other enterobacteriaceae, and you've now appreciated they don't just cause gastrointestinal issues, they can cause other complications depending on how it's disseminated, can cause UTIs, and cause bloodstream infections such as sepsis, can cause even meningitis, pneumonia, depending on how the diagnosis and the route of transmission. So I believe you're now quite familiar with some of these pathogens, and today we're going to look at Salmonella and Shigella. So starting with Salmonella, first of all, this is an electron micrograph, as you can see on the screen, and you can see that one major characteristic feature is its flagella. I believe most of the other pathogens that you've looked at, you see either they have a single flagella or a double flagella, but you can see this Salmonella itself has several or quite a number of flagella, and we normally call this petricious, and very important, a very good virulence factor because it enables this pathogen to move all the way down to the gastrointestinal tract, past the stomach, and establish infection in the intestine. So it needs a very good mechanism to move from from each site to another. So, and that's a very good virulence factor the pathogen possesses. So when you look at Salmonella, just as an introduction, it causes a disease, and which is something you're quite familiar with, typhoid, and can also cause non-typhoidal diarrhea. And it's one of the major causes of foodborne diseases that causes gastroinfections. And This pathogen, if food is not handled properly, can be introduced into the unknowing host. So food has really have to, if we are trying to prevent it, we need to have prepare our food in the best hygienic standards as possible. People who are also preparing food, particularly restaurants or in schools or in other settings, also need to be vaccinated. So this is one of the real diseases with a vaccine. That's a typhoid vaccine that should be given to and given boosters regularly. Food handlers should also have a certificate to make sure that they've been vaccinated to avoid passing on this diarrhea-like infection. Again, this pathogen infects both humans and also many domesticated animals. So it's really important when preparing food to make sure that you have a clean environment, cook your food thoroughly, and observe strict hygiene measures as you prepare the food. So let's look at some important characteristics as an introduction of the Salmonella. Again, as you can see, the common theme of these lectures that you have, these are gram-negative rods, as you can see. They're motile, quite motile. Again, I've introduced to you the flagella, known as the petricious flagella, meaning that the flagella is distributed across these rods, again, which gives this pathogen ability to move from one cell to another as it goes through all the way through the gastrointestinal tract or to colonize the intestinal cells, where it will be able to thrive, obtain nutrients, but cause just detrimental effects such as diarrhea. For us in the lab, it's important to be able to differentiate these gram-negative rods, and we normally look at the different factors that it produces, either when it's consuming glucose or consuming other nutrients. First of all, it does not ferment lactose. I'm sure you've been introduced to pathogens, these gram-negative rods that ferment lactose and produce different color changes, but it produces hydrogen sulfide. Again, a very
Excuse me, Dr. Marianne. I think you've muted yourself. Hello? Hello, Dr. Muridi. We can't hear you. I think you've muted yourself.
now. Okay, I'm hoping you can hear me. My connection went up and down, but hopefully this is now more stable. Sorry for that. So we're looking at important characteristics, and one of them I've already mentioned, 2 gram-negative. They're quite motile and do not ferment lactose. So again, that's important for us when you're trying to look at lab diagnosis, and I'll take you through the importance of this. Also, as it breaks down the glucose and other nutrients, it produces hydrogen sulfide. Again, this is important to us as it breaks down its nutrients, the production of hydrogen sulfide, which again can be identified in the lab using different chemical reactions. Again, these chemical reactions, we see the changes in the agar plate where it darkens. Again, a very important characteristics when it comes to the diagnosis of this pathogen in the lab. Another important characteristic, they are facultative anaerobes. And just as a reminder, facultative anaerobe is any organism that makes energy by aerobic respiration. They can thrive or produce energy if oxygen is present, but also are capable of switching to ferment. They're also capable in production of energy using other mechanisms such as fermentation if oxygen is not present. So they're quite versatile, depending on the environment they're in, that they can survive with or without the oxygen. Another important characteristic is the availability of different antigen factors. These antigen factors are important for us, again, in terms of identification. When you do some simple ELISAs to identify, we see agglutination, that is antibody-antigen interaction. And these antigen factors are normally termed as O, which in other textbooks can be the somatic antigen. We have the H antigen that is found along the flagella, and the VI or virulence antigen that is also found around the capsule. So these again important factors that this pathogen possesses, and these factors help us identify them in the lab, and two, also advantageous to the bacterium in that it increases its antigenicity, so attracts other cells towards it in terms of energy requirement, in terms of obtaining nutrients. Flagella, I'm sure you're quite familiar, the importance of flagella, moving it from one area to another. And again, in the next slide, you'll see the different virulence factors that I've already introduced to you. I've already introduced the O antigen expressed on the cell wall and the capsule. Again, I've introduced to you what antigens are, but also this antigen or the somatic factor also prevents this pathogen from being digested or through phagocytosis that is mounted by the host immune mechanisms. So the O antigen is very crucial again to the bacterium. I've mentioned to you the H antigen is also a somatic factor that is expressed on the flagella. Again, the importance of flagella is helps it move, so its motility, and also the H antigen not only boosts its motility, but also helps it adhere to different cells as this pathogen swims or moves across different cells. So it's very important for the flagella to have this H antigen, which helps it in adherence and also contributes in its resistance towards any immune host mechanisms mounted, such as phagocytosis. I've also mentioned the VI somatic factor or antigen that is expressed also in the capsule. Again, just boosts this pathogen's ability in evading the immune system and also inhibits complement binding, which also factors the host mounts to prevent this pathogen from growing and also in terms of destroying this pathogen. So all these somatic factors, the VI, the H, and O are very crucial to this pathogen that we're learning about today, and that is the Salmonella. You can also see that the Salmonella produces several toxins. Again, the toxins are important because either they contribute in the destruction of different cells, and you're going to see some of these cells are cells that produce the mucus that lines the different intestinal walls. It needs to burrow through or dig through this mucus layer to be able to attach itself to intestinal cells, so it can obtain different nutrients. It produces different toxins such as cytotoxins. It produces even LPS or the endotoxins, and you also have enterotoxins, which then irritates the intestinal cells or in the intestinal membranes, which then starts the cascade of diarrhea symptoms in this patient. It also has other different factors. Again, I'm going to send you this diagram and several notes, such as fimbriae. Fimbriae is also important. These structures, again, helps this pathogen adhere to these particular cells, such as the intestinal cells, which are lined with the mucus, so quite slippery. So it needs all these factors to be able to attach themselves, adhere, and then instigate that colonization, obtaining nutrients, and again, with the toxins it produces, causes all these other symptoms that we see. So each virulence factor or each component of this pathogen is quite useful, the way they've evolved themselves to be able to thrive within the host so that it can obtain its nutrients, reproduce, etc. But all these virulence factors also cause the symptoms that we're going to see shortly. So having that in the back of our mind, looking at Salmonella, I've already mentioned to you, causes two major diseases, which are normally termed as salmonellosis or salmonellosis, as you pronounce it more slowly. And the major disease here, we're looking at enteric fever, or in other words, typhoid. And this typhoid is classified as enteric fever, resulting when this pathogen not only invades the gastrointestinal tract, but it's able to invade also the bloodstream once it starts its colonization, causing enteric fever. It can also cause acute gastroenteritis. This is results when you've also consumed this pathogen, which then irritates the intestinal cells, causing the foodborne infection or intoxication, again, leading to diarrhea. So your patient can display diarrhea-like symptoms, or your patient can exhibit both diarrhea-like symptoms accompanied with fevers and other discomforts, which can then be termed as enteric fever or typhoid, that will need more investigation and a different route of treatment. So I mentioned to you, we can either have typhoid, which is characterized by the seeping of this pathogen into the bloodstream, and non-typhoidal, so causing gastroenteritis or stomach upset. And the patho subspecies that have been implicated to cause typhoid fever, it's either known as typhoid fever or paratyphoid fever. We have Salmonella typhi, these are the subspecies, or Salmonella paratyphi A, B, C, depending on the different serotypes and serovars that it is displaying on its cell wall. All the other Salmonella serotypes normally have been implicated in causing simple gastrointestinal discomfort, diarrhea, that either the body can clear after 2-3 days, or in case if it's severe, we need to introduce some antibiotics to help achieve the treatment. So when you look at the burden of disease, I've mentioned to you, we have the invasive disease that seeps through the bloodstream. And I've mentioned to you, this is just a summary of what I've mentioned in the last 2 slides, where we have the typhoid itself, where we see that enteric fever. Then we have the non-typhoidal gastroenteritis caused by Salmonella. And I've mentioned all the other species of Salmonella, such as Salmonella enteritis, Salmonella typhimurium. All these have been implicated to cause gastroenteritis, which is very common. You can see globally, we normally see several millions of cases, almost 100 million cases worldwide. And if not treated well, also causes death. And we also, of course, we have the typhoid fever characterized with the different symptoms that you're going to see. Again, very prevalent, particularly in our region, in sub-Saharan Africa and also in Asia. And it's fantastic that we have a vaccine that has helped us reduce these numbers of typhoid fever. And of course, the non-typhoidal salmonellosis, I've already mentioned again, is also quite prevalent. One of the major causes of diarrhea, particularly in our continent. And again, this needs some serious investigations, and there's quite a bit of implications on how to move forward in terms of treating diarrheal diseases. One fantastic thing I've seen, or a positive effect of COVID, and I know that is like an oxymoron what I've just said, but we've somehow in the last 1.5 years, we have seen a reduction of diarrheal diseases across different age groups, in not only in our country, but also in the continent, the sub-Saharan continent, and even in the world. And really what has contributed to this is that, first of all, we are now very conscious in terms of hygiene. We are washing our hands even more thoroughly than before. Again, this is one of the measures our Ministry of Health and different organizations are telling us to do in terms of preventing COVID-19 infections. And that has had a very good side effect in that we are seeing less and less diarrheal infections in our children and also in areas that were quite endemic in getting these infections. So again, I'm hoping that once we're all vaccinated and we're told we can now live our lives without fearing COVID, we'll continue these measures of observing strict hygiene standards, such as washing our hands frequently. And again, this will help us reduce the real infections that you have seen in the last 1.5 years of what has happened. So let's just look at how, and this is quite obvious, the different routes of infection. My slides. Okay, so you just have to go the way it's presenting to us. So we've introduced the pathogen, the different diseases, both typhoid and non-typhoidal. Looked at enteric fevers, that is caused by, in other words, typhoid, and gastroenteritis, that is caused by the non-typhoid causing type of Salmonella. And the major routes of transmission of both is through fecal-oral route, really, when you don't observe strict hand washing measures. And this can be either be person to person, so spread by a chronic carrier, so someone who's probably infected and is displaying symptoms, but yet goes ahead to prepare the foods. Then we have also food that is also contaminated. So we've used dirty water or food that is already contaminated with a salmonella. So these are different ways that this food is introduced. A century ago, we had a very, there's a story of a chronic carrier, and her name, I don't know whether you've heard about it. I think there's even a movie known as Typhoid Mary. This lady was in London. She used to work in restaurants, and wherever she went to work, or wherever she was employed in London cities, in the restaurants, there would always be an outbreak of typhoid, or several customers complaining later that they had typhoid. And from their investigations, they realized the common thing all these restaurants had was this lady called Mary, and I guess that's where her name came from, Typhoid Mary. And they tried to take away her license, stopped her from working from restaurants, but she'd change her identity, start working another restaurant, and cause this chronic or this outbreaks of typhoid. So chronic carriers are something we need to be aware of, and I think that's the most common chronic carrier that is documented in history. There's a lot of publications and literature that alludes to Typhoid Mary. But anyway, that's a short interlude. So once this pathogen is introduced into your system, either from the routes I've already mentioned, we see that it swims through the gastrointestinal tract and can take 7 to 14 days before we see any symptoms. So as it swims through this and passes through the acid-rich environment of the stomach, it takes the form of it becomes inactivated, does not burn any energy, and it's able to evade the acidity of the stomach lining area, and quickly, successfully is able to swim through to the intestinal region, again because of its quite powerful or strong flagellum. And with that, it's able to burrow through the mucosa that lines the intestinal tract. And again, using the several toxins that I've mentioned, it also uses and not just the toxin, the fimbriae, and other factors to be able to adhere to the mucosa of that lines the gastrointestinal tract. Once it's able to line there, it's able then to obtain its food, obtain nutrients, and also colonize the system more. And with that, it's also able to invade different cell types along the intestinal cells. And with that, it's able to seep through the mucosal layer, the intestinal layer, seeps through to the mesenteric lymph nodes. And once it seeps through the mesenteric lymph nodes, gains access to the bloodstream. And once it's inside the bloodstream, that's why you see your patient displaying different symptoms such as fevers, nausea, stomach cramps. By then, they should also already started exhibiting some of these symptoms. And on top of that, the salmonella is also able to seep through the different organs. And the most common organ particularly if you're a chronic carrier, is the gallbladder. So chronic carriers may not, at some point, may not even display some symptoms, but are chronically carrying this pathogen in the gallbladder. And I guess that's where we see the story of typhoid Mary coming up, and several other people who are known to be chronic carriers of this salmonella, and reinfection occurs again through the excretion of the bile, which then ends up in the feces. And this route of transmission continues over and over again. So depending on the pathogen, if it's a typhoidal salmonella or non-typhoidal, again, your patient will display different symptoms. So when you're looking at gastroenteritis, you see that your patient again will either will display diarrhea-like symptoms. There'll be some vomiting, abdominal pain, that nausea feeling, and diarrhea. And if this is not well treated, your patient may become dehydrated, leading to hypertension. So this has to be treated in good time so that you're able to rehydrate your patient. Again, if your patient exhibits either persistent or high fever, again, this may suggest bacteremia, where this pathogen has been able to seep through the bloodstream, causing this infection. So this is some of the most common symptoms that I've mentioned. So we have the enteric fevers, and we have where it seeps through the bloodstream, causing septicemia, and the different pathogens that cause this. Again, this I introduced to you in the last few slides, and I've mentioned the carriers again who are symptomatic, who carry this pathogen without displaying any symptoms, and the gallbladder is normally the reservoir. And the most common subtype is the Salmonella typhi, which then again is excreted in the bile, which then goes into the feces, and hygiene measures are not well in place, and then be introduced again through the fecal-oral route. So the patient may display any of these symptoms, from general tiredness, fevers, diarrhea, vomiting, stomach cramps, and again, it's important to be able to take this patient's history and again obtain the correct specimens to help you make the correct diagnosis. So the specimen here, you'd either collect a stool sample, if that is, you can obtain that. For enteric fevers, you may even collect some blood, do some blood cultures, or even bone marrow cultures. Again, it depends on the symptoms your patient is displaying. But the most common specimen that we would normally take back to the lab is a stool. And once you get the stool in the lab again, you first of all start off with your macroscopic investigation, what you see with your naked eye, before you progress to do your microscopic, where you then culture and look under the microscope to see whether you can identify this pathogen. So with the stool specimen, we normally first on day one, culture it and enrichment media, so that you can make sure that the pathogen of interest can be obtained in large quantities. Remember, stool specimen will have both we have several bacterium. Again, some bacteriums are beneficial for the pathogen and some are not, and we really want to help identify the disease-causing pathogen. So either culture it first on day one an enrichment media, which then selects or is beneficial for the pathogen of interest. And here is the Salmonella. So use enrichment media will have selenite F broth, which the bacterium would need for its nutrient requirements. So once we have done that and we have colonies, the second day we do another subculture, either on now specific or specific agar or culturing mediums that selects for Salmonella or the pathogen of interest. And here we have several agar plates or agar mediums that we can use. The most common one is the Salmonella Shigella agar, or SS, that we normally use for this isolation. Again, we incubate it for another 18 to 24 hours, before we see our colonies. And one thing to note here, our colonies will either have these dark spots, and this is because of the production of hydrogen sulfide that it produces as it obtains the nutrients and as it ferments some of the sugars and nutrients that it requires for its growth. So that's a good indicator that you may, your patient may be infected with Salmonella. Other tests that we can use in the lab, we normally employ some biochemical tests, and I believe the TSI test has been introduced to you. And again, if not, I know you have a practical this afternoon where you'll be able to you see this in practice, where we use this slate or this slant or this agar and culture our subculture our colonies on it. We want to identify that it's Salmonella, so culture it here, and then we normally see different color changes depending on again, the production of different gases as it ferments or uses the glucose or different sugars for its nutrient needs. So in case of Salmonella, remember it produces hydrogen sulfide as it ferments. So you'll be seeing a very darkened butt. So that at the edge of this TSI, the TSI test, you see a darkened butt. Again, this is because of the production of hydrogen sulfide. And this is important for us in the identification of several of these of the Salmonella species. Again, I believe this is I'm taking you through this theoretically, but I believe once you do this practically, you'll be able to appreciate the different color changes on the TSI medium. Other tests that will be introduced to you is the urease test, where again, this Salmonella lacks this enzyme, the urease, which is important in breaking down some of the urea or nitrogen factors. So it lacks that enzyme. So if you do an urease test, it will be negative. So in short, when you culture on your Salmonella agar plate, Salmonella Shigella agar, as depicted on this diagram, to help you identify your colonies, you'll mainly see these darkened spots where the colonies are growing, and this again is due to the production of hydrogen sulfide, and this is a very good indicator or important factor in terms of lab diagnosis. Other tests that have been in use, but they're rarely used at the moment because of more sophisticated tests, such as molecular tests, where you do some PCR on your specimen. Again, I remember mentioning PCR is 99.9% accurate because it detects these pathogens, the DNA. So apart from the molecular, apart from the culturing, those are older tests known as the Widal test. Again, the Widal test would normally detect these antigens. Remember the somatic factors O, H, and VI. Remember, VI is found on the capsule, H factor is found on the flagellum, and also O is found on the cell wall. Again, all these are antigens. And using the Widal test that I've displayed here, where they employ the antibody antigen agglutination test. So these factors or antigens are present, you see a color change on your Widal test. Again, most clinics, most labs are facing out this Widal test, but it's in use. Again, very useful in helping us identify this pathogen of interest. Okay, so looking at treatment for gastroenteritis, as I mentioned, for most of us, healthy adults, and even some healthier children, it's usually self-limiting, again, without any treatment. What you need to watch out for is the fluid loss. So make sure that there's a balance to that. And also your doctor, your physician may prescribe something to control the nausea, vomiting, or general pains that you may experience. And in case of severe infection or in case of enteric fever, different antibiotics again can be prescribed. Again, this prescription, the person prescribing or the center prescribing should really adhere to the hospital antibiogram, where again this resistance that may have been detected, and the correct antibiotic should be used to treat this pathogen. But most of the time, the issue is self-limiting, and what you need to take control of or hold of is the loss of nutrients and also fluids, fluids and electrolytes, which may lead to dehydration. Okay, so in terms of prevention, apart from treatment, I think we are quite now aware of observing very strict hygiene measures. And this I pulled again when I was reading the story of Typhoid Mary. They had this poster everywhere in the 1800s where they were trying to educate people about the typhoid because of the outbreak they were seeing in London. So I thought this is a really good tool they used at that point, where you, again, make sure your food is well prepared, the foods and all these things, that there's strict sanitary measures taken. But now, as I mentioned, we have vaccines that are available. We have different types of vaccine. We have one of the vaccines that we use is a killed version of the Salmonella typhi, administered by a shot. And also in our country, we also have a live attenuated vaccine, again, that helps us deal with typhoid fever, and is normally taken by mouth, which is quite effective because it starts eliciting an immune response right at the source of transmission from the mouth all the way down the gastrointestinal tract. So we find some antibody factors elicited by the vaccine, which again boosts your protection in getting this typhoid and also transmitting it to the next person. Again, it's an important vaccine, normally recommended, and in our country, in our city, it's compulsory for any food handler to have an up-to-date certificate showing that they have gotten this typhoid vaccine. Other vaccines given to food handlers that may have been mentioned to you, we have the cholera vaccine that is normally also quite effective in preventing Vibrio causing diarrhea. I know we also have the rotavirus vaccine, but that's, I think, I'm sure you covered under when you're doing virology and you're looking at rotaviruses. So again, this is a win for us to have vaccines. Again, I keep mentioning we have a problem with the antibiotics. Again, we have a very high rates of antibiotic resistance. So we can have proper vaccines and a vaccine that elicits responses, a strong enough response without needing of a booster, that would be quite helpful. So moving on, so I'll cover both Salmonella and Shigella, then we can allow for questions before we quickly look at Pseudomonas, which is in a different class. Shigella as well is one of the real causing pathogens. The slides are not moving through, but again, as you can see, in my introductory slides, I think you appreciate the discomfort that this pathogen may cause, such as diarrhea, vomiting, and just gastrointestinal discomforts. And when you look at this, you're looking at gram-negative rods, and you can see again a distribution of its flagella, which gives it a good advantage or capability to be able to colonize the gastrointestinal tract. My slides are stuck. Okay. Okay. I don't know where my slides are misbehaving. I don't know why it does that. So again, important properties of the, let's start again. Important properties of the gram-negative, of this gram-negative bacterium. And now we're looking at Shigella. We've looked at Salmonella. And here I'd like us to note a few differences between the two because they'll help us make some diagnosis. Remember, Salmonella I've mentioned the production of gases, the availability of the petricious flagellum, and several others. It's motile. So let us look at the Shigella bacterium because more or less they cause similar diarrhea, but there are several differences towards them. So first of all, again, they're both gram-negative rods, as many as the theme of most enterobacteriaceae. One thing, they're non-lactose fermenters and do not produce gas from any of the fermentation. So again, if you're trying to make a diagnosis using the TSI or looking at gas production, there'll be no gas production from the fermentation. So again, we do not there'll be no presence of hydrogen sulfide. So their colonies will not also have that blackened spot. The TSI medium, again, when you look at the slant, will not have a darkened spot as well. So that's an important factor to differentiate between Salmonella and Shigella. Another important factor here to introduce to you, they're non-motile. Sorry for the, I mentioned it could be motile, but Shigellas are non-motile, while Salmonellas are motile. So they require other mechanisms to move from within the gastrointestinal tract, cause infection. They have similar somatic factors, but here you find that they'll have the O somatic antigen on their cell wall. They'll also have the VI, but will lack the H again because of the lack of the flagellum. And they're known to cause bacillary dysentery. So remember, we've looked at typhoidal or enteric fevers caused by Salmonella. Shigella is known to cause dysentery. And dysentery, Lily, is, in other words, is bloody diarrhea or diarrhea that contains some blood particles. So that's something also to something important for us to be able to help us identify the different types of diarrhea. I don't know whether you've done cholera. You'll see that cholera also, the diarrhea caused by the Vibrio pathogen that causes cholera. The diarrhea is different in color. It looks like rice water or quite whitish in color. And that again is a good indicator of the Vibrio pathogen. And for here, for the Shigella pathogen, we see that the diarrhea has some blood spurts or some bloody diarrhea, and that's why in most textbooks it causes diarrhea caused by Shigella as bacillary dysentery. Again, my slides are taking too long to move from one slide to another. And with that, Shigella is divided into different subgroups. We have group A to D, and within these different groupings, again, grouped because of their serotype availabilities, so that these are serotypes found on the cell wall, using simple ELISAs in the lab, antigen antibody agglutination techniques, we've been able to identify different serotypes. We have group A, where we have the Shigella dysentery. Then we have group B. These are the different Shigellas that have been described. And the major one here, Shigella vaccinari. We have Shigella boydi or Shigella sonei. Again, all these fall under the Shigella group of families or Shigella species. And all these have been implicated to cause the bacillary dysentery. But the most common Shigella in our region, this is where we are concentrating in sub-Saharan region, is the Shigella flexneri that causes this distressing diarrhea, and it needs to be treated and in the correct format. Again, there's quite a lot published both by WHO and also the CDC regarding Shigella flexneri. But our take-home message here, I just wanted you to highlight, out of the 4 groups, if you're working in the lab or you're dealing with patients with who are infected with Shigella, more or less in our region, they'll be infected with the Shigella flexneri, which is quite prevalent in contaminated foods or water. So again, pathogenesis of this, it's quite infectious, very, very infectious. It requires just a very low infectious dose to be able to cause the infection. So the higher the dose, so the higher bacilli or the rods that are available in the foods or the water that you're taking, the bigger the dosage, the more lethal it can be. So we really need to observe strict hygiene measures to avoid this infection. So it's very contagious, and you need to just ingest a few of these bacilli to be able to see this infection. Again, I've already mentioned Shigellosis, the diseases caused by Shigella. The most prevalent is bacillary dysentery, and this is the passing of blood and also mucoid in the stool. So this is a good indicator. There's something irritating your colon or the intestinal membrane that's causing this bleeding, and this is the toxins produced by the Shigella, and this can be transmitted, excreted in the diarrhea. The incubation period is quite short. Within a day to maximum 3 days, you'll be able to see this watery diarrhea that is bloody, accompanied with a fever and different gastrointestinal discomforts. And again, the bloody streaks or bloody diarrhea is a good indicator that you may be infected by the Shigella. And I've mentioned that the toxin that the Shigella produces, and then one of the major toxins, the Shiga toxin, that's enterotoxic, and again causes, as different textbooks would describe it, it's neurotoxic and cytotoxic, meaning that it's able to destroy or cause destruction of the cells and the mucosa membrane that lines up the intestinal tract as the pathogen burrows through to cause or establish that infection. It's through that and the changes instigated by this toxin that leads to the bleeding or around of that area of the intestinal tract, causing, which seeps through the diarrhea or the gastrointestinal tract, causing these changes, both the watery diarrhea and accompanied by the bloody streaks. Again, the Shiga toxin is implicated in this. And really, the early stages of the infection is where you first see the diarrhea, again, which the toxin contributes to because it causes how these nutrients are being absorbed to change. So the water leads to a bigger loss of water and nutrients. And then if not treated, or if you've had a bigger dose of this pathogen, you've been exposed to a much higher dose of it, it will then lead to the destruction of the tissues and the cells that line this intestinal membrane, again causing the dysentery or the blood streaks within the diarrhea. And of course, I mentioned your patient may have some fevers, may also have some gastrointestinal cramping and pains, again, still attributed to this toxin that the pathogen produces. So you can see that it causes quite a bit of damage in the colon area and in the gastrointestinal tract area, where it burrows through and causes this bleeding, which then leads to the dysentery. So I mentioned most people who are, this is just a summary of the signs and symptoms, will have the diarrhea, fever, and stomach cramps. Again, they start quite early, following exposure within a day to maximum 3, and the diarrhea is usually quite bloody. Again, as much as it sounds quite sinister, most people are able to clear this infection. Again, just making sure that this patient is well rehydrated. We don't want to see a blood loss or nutrient loss. So this needs to be taken into account. But if we are treating a child or an elderly patient, we may need to intervene with appropriate antibiotics to be able to help us get rid of the diarrhea. So I mentioned several of these symptoms: the blood, the mucus, the abdominal cramps that may be accompanied by this infection caused by the Shigella. And the most important virulent factor it has, apart from the different ones that we mentioned, the capsule, the fimbriae that helps it attach, are some of the toxins it produces. And one of the most common toxin is the sugar protein, sugar toxin. So let's look at, in terms of diagnosis, again, your patient will have all the symptoms. So first of all, once you take the history, you may want to take some part of the diarrhea specimen. So you'd want to, first of all, when you do your macroscopy, you need to note, is it mucoid? Is it bloody? Again, you can take the diarrhea specimen, or you can do a rectal swab in case you're dealing with a young child or someone who's elderly. In case, again, here's just a point of concern. If the lab is quite far from where the patient is, you can transport this specimen using the Curry-Blair transport medium. This medium is good because it's able to maintain the bacterium because we want to be able to diagnose it once it comes to the lab and identify it. We need to have a bacterium that is thriving within its the real specimen. So using Curry-Blair really promotes the thriving and the survival of this pathogen. Again, I've mentioned once it's in the lab, I would want you to do a macroscopy where you identify with your naked eye different characteristics of that diarrhea. Again, that really helps us in making the correct diagnosis. You can have a watery diarrhea, a bloody diarrhea, you can have a diarrhea that looks like rice water, you can have a darkened diarrhea. All this is very crucial because it helps us identify the pathogen. And here we are dealing with Shigella, so we'd expect to have some bloody streaks or some blood within that diarrhea. Again, you could do a microscopy, but I'd really want us to move to the culturing, because with that, we are able to identify the species, the specific specimen, and that with that specimen from the colonies, you can do the microscopy, and we'll normally either use the McConkey or the Salmonella Shigella agar. Again, this will be, you'll be able to see this practically within the lab once you do your practicals. And if you're using the McConkey, which promotes the fermentation, we see non-lactose fermenting colonies, so there'll be no color changes within the McConkey. So observe non-lactose fermenting colonies. And with the TSI test, when we use that, when you look at the medium, we'll see that, first of all, there'll be no gas production or any color changes as we had observed with the Salmonella, so we no darkening of the butt, but we'll see an alkaline slant. Again, this will be more clearer to you when we do the practicum. And again, I'm going to take you through that at the end of the lecture on the different characteristics on the TSI medium. Treatment, I've already mentioned, make sure we are replacing and taking an eye or keeping an eye on the fluid and electrolyte loss. So making sure that this is well replaced, but many cases, fortunately, are self-limiting. But again, there are several antibiotics that can be given, depending again on the guidelines of the different setting of where you are based in your clinics or in a hospital setting, and depending on the antibiotic resistance that is noted. At the moment, we do not have any vaccine that is licensed for Shigella. I know there are few that are that are being developed. So in phase 1 and phase 2, we really need to have a vaccine. But again, I cannot emphasize enough, we're in an era of antibiotic resistance. So we need to be able to prevent these infections instead of concentrating and treating them. And one of the best preventative measures is a vaccine. And I'm hoping that these vaccines will be licensed soon to help us deal with the rise of this antibiotic resistance. But with that, we also need to observe strict hand and also food handling hygiene. So the frequency of hand washing really helps, and that has been evidenced by the last 1.5 years. We've been washing our hands quite frequently because of COVID-19. We've also seen this diarrhea, bloody diarrhea incidences quite drastically reduced in a hospital setting. So we really need to adhere to that. Of course, we need to have the best practices in how we handle our food and vegetables. So again, I'm sure within your setting, you're also able to handle your vegetables well to prevent consuming any of these products that may be infected with any of these diarrhea-causing pathogens. So in summary of both Salmonella and Shigella, we've seen that Shigella, the main reservoir is actually humans, but for the Salmonella, we've seen humans and also domesticated animals, even their products, such as eggs and so
Yeah, Nicolia was great.
Sorry?
Don't decrease excess. Don't
Hello, please mute if you're not asking a question. Kindly mute if you're not asking a question. So here we're just summarizing the two pathogens. Infectious dose. So Shigella we have seen is quite infectious. So you require just a small dose for you to start exhibiting symptoms. Where Salmonella, you may need to consume a quite bigger dose to see some of the more characteristic symptoms. Of course, diarrhea is a prominent feature in the Shigella and also in Salmonella, but for Salmonella typhi, that is implicated in causing typhoidal infections, you may see other symptoms such as fevers. And then invasion of bloodstream, mostly the Salmonella has that capability, which then we've already discussed enteric fevers, septicemia and bacteremia. For Shigella, we don't see the invasion of bloodstream, but it causes enough damage within the gastrointestinal tract. So that's why we see the bloody diarrhea. Chronic carrier state, you've seen Salmonella typhi. I mentioned typhoid Mary as a good example. So you can be a carrier, and they've shown that the gallbladder is where this pathogen can remain in an asymptomatic state for quite several years if not well treated. In terms of identification, we will look at different fermentation, such as lactose fermenters and the production as it consumes, burns energy, the production of hydrogen sulfide. And if you want to differentiate between the two, you see that the Salmonella species, hydrogen sulfide, produce hydrogen sulfide, and most of them produce hydrogen sulfide, while Shigella strictly does not. And I've mentioned vaccines availability. You see that we have a vaccine towards typhoid, but no vaccine as yet towards the other Salmonella, which again would be important to have, or the Shigellas. Okay, next slides. Keep jumping from one slide to another. So with that, let me look at some of the questions you have before I introduce to you this other pathogen known as pseudo, or in full, Pseudomonas. So from one of the questions, I can see, can our bacterial GCAT be pushed to August, please? Okay, that one is more administrative, so we'll look at it. Salmonella typhi does not produce gas, so there'll be no blackening of the butt. Again, that is from Marcus. So Marcus also says we are talking about Salmonella Shigella typhi was noted as Oh, you didn't tell me I'd muted myself. I'm just here talking to myself. Sorry for that. Where were we? Where did I lose you? Again, with my slides moving up and down, it means I'm pressing different keys. I can see why is Shigella not non-mortal, and yet it had flagella in one of the slides. I actually corrected that slide and said that Shigella do not have the flagella whatsoever. What they have is fimbriae, which helps in the attachment on these cells. So it does not have flagella, it's non-mortal. What it has are the structures known as fimbriae that are distributed across this rod-like bacilli, which again helps it in its attachment. So it's non-mortal, so uses other mechanisms such as adhering to one host cell to another as it moves along the gastrointestinal tract. So I am hoping I've answered your question in terms of hydrogen sulfide. And that's the production of hydrogen sulfide is an important ability of the different Salmonella and it's very important to us, particularly when we are screening and identify Salmonella in the different selective mediums. But we have some Salmonella that do not produce this hydrogen sulfide, and these have recently been identified actually. And one of the Salmonellas that have been implicated not to cause or not to produce this hydrogen sulfide are one of them that has been described is Salmonella enterica. And again, I'm going to just send these notes to see how Salmonella is evolving not to produce this factor, which helps us in identification. And this is identification we normally use when we use a different selective medium, and also when you use the TSI standing for triple sugar ion agar, which helps us identify different ranges of Salmonella that are negative towards in the production of hydrogen sulfide. So the ones that have been identified, we have Salmonella enterica that has all the characteristics of Salmonella but does not produce hydrogen sulfide. We also have Salmonella typhi or Salmonella in full Salmonella typhorium that also does not produce the hydrogen sulfide. There are several more that I'll send to you. I've seen in literature, there's Salmonella Kentucky, Salmonella Heidelberg, Salmonella Delby. All these have been discovered in the last 5 years or so, again, because of we've improved our molecular identification techniques, etc. So this is an area that is evolving, but out of the Salmonella that are important to us, there are some that produce this hydrogen sulfide. So allow me to introduce to you Pseudomonas. And I don't know why my slides keep jumping up and down. So you're moving on from Enterobacteriaceae, and I just want to mention a different type of pathogen known as Pseudomonas. And the full word, we normally call it Pseudomonas aeruginosa. And this is a Latin word, aeruginosa, meaning quite colorful, i.e. the colonies that are produced by Pseudomonas aeruginosa, or this bacterium of interest today, are quite colorful. They have different colors. They could be green, they could be blue. And again, it's because of the different enzymes and how they utilize the agar and the glucose found in the agar produce these different colors. And they also produce a biofilm, which is important for us in causing different types of infections. They produce different pigments, and these are the pigments that contribute to its color. And also, apart from the colorful colonies, they also have a very fruity-like smell. So if you're trying to identify a specimen that you have grown on your agar plate, such as McConkey or different agar plates that you're using, first the color change. These are very colorful bacterium, or the colonies will be quite colorful, and also will have a very fruity, some textbooks describe it as sweet grape. For me, when I culture it, yes, there's a distinct smell. How they came to conclude that distinct smell is fruity, maybe it depends on how you smell it. So that's a very good indicator for us to identify Pseudomonas aeruginosa. They are aerobes, so they thrive in areas that are oxygenated to be able to produce energy and burn up ATP. And mostly you see they cause opportunistic infections, particularly in hospitalized patients. Patients who either have a catheter or have an equipment attached to them. Remember, this pathogen produces biofilms, which helps it adhere to non-living products or innate products or organisms. And that's why you see that the Pseudomonas may be in a catheter, maybe in a pipe, or maybe in an intravenous tube, which is then introduced in your hospitalized patient and causes opportunistic infections. They are rod-like, gram-negative rods, and that's where we group them together, Enterobacteriaceae. They contain this flagellum. They do not form spores. And for us to identify them, apart from the color, the smell, you can do different biochemical tests. You see that they have this enzyme, catalase, and also oxidase, and they'll be positive for these enzymes. So I've mentioned they produce color, so it's a very good indicator for us. So they have different pigmentation, and the smell will be quite distinct. And for your patient, if you want to see if your patient, or a good indicator that your patient is infected with Pseudomonas aeruginosa, you can look at the patient, either the pus, if it's a wound that's been infected, or any other product. But mostly we look at the pus. If you're dealing with a wound, you see that your pus is not your usual creamish color. It will have a different color change, like blue greenish pus, which is an indicator maybe perhaps your patient may be infected with a Pseudomonas aeruginosa. And again, another thing to note about Pseudomonas is a huge antibiotic resistance, or it's broadly antibiotic resistant. So really we need to pay close attention on, particularly patients who are immunocompromised, patients who are either undergoing through chemotherapy or have been hospitalized for quite a while. They have different tubings or innate mechanisms to help them relieve their pain, or catheters. So we need to make sure either we are changing them frequently, or we are watching out for any of these infections, because remember I've mentioned it forms biofilms, which helps it adhere to this innate product. So we really need to be careful in terms of this opportunistic infection. You can see that if you culture it on your agar plate, you normally see colorful colonies. There's a colony here that you see has a blue greenish pigmentation. Again, a very good indicator. It's one of the few bacteria that has its pigmentation changes. And again, you find it in the environment, again, because of the production of these biofilms that helps it adhere to these non-living products. And I've already mentioned to you, it's very common in hospitalized patients who causes nosocomial infections. So it has, as you can imagine, different virulent factors. I mentioned it's already motile. It has a fimbriae that helps it adhere and attach itself to different products. And on top of that, it has this biofilm. Again, it's an important characteristic of Pseudomonas because it's able not just to adhere to living products, but non-living products, again, which is detrimental to any hospitalized patient that is attached to an equipment or an innate product in terms of their treatment plan. It also has different enzymes. I've already mentioned the catalase, the oxidase, and also it has different proteases, hemolysins. It helps it lyse different cell types as it continues to get its nutrients. And of course, it has enzymes that contribute to its different pigmentations, the different colors that it produces. In terms of infections, it causes a wide range of infections depending on how it's been introduced into the living host. For example, it can cause skin infections if either you're using a swimming pool or a jacuzzi that is not well treated. Remember, it produces biofilm, so it will attach itself to some of these equipments then introduced to you. In case on the skin area, if you use contact lenses, again, the lenses can be contaminated by this Pseudomonas aeruginosa. causing this type of an infection. You can see that pigmentation, the greenish color, again attributed to most likely that this patient either is a contact wearer or has been exposed to the Pseudomonas species. You can see folliculitis again that can that is attributed again to the Pseudomonas species. So other infections, depending on how it's been introduced, can cause a UTI. Again, if your patient is using a catheter, it can cause pulmonary or infections if this patient is intubated or has some tubings that are assisting them to breathe. If your patient has a wound and you realize the wound is producing pus that has a distinct color, again, it makes you suspect Pseudomonas aeruginosa. So it causes a wide, it has an extreme broad host spectrum, again, depending on how it's been introduced. Remember, it's an opportunistic pathogen. So again, we need to watch out, particularly in patients who are immunosuppressed or have immunodeficiencies and have different medical products or devices that are attached to them, again, to prevent this opportunistic infection on your hospitalized patient. So lab diagnosis for Pseudomonas aeruginosa is usually quite obvious. First of all, just take the history of the patient, look at the site of infection. So if you're dealing with a wound, you probably want to do a wound swab. And from there, you may need to look at the color of the pus. If you're dealing with a UTI, again, collect the urine, note the smell, note the different color changes. Again, all that will indicate to you if you're dealing with Pseudomonas aeruginosa. Again, you can quickly culture on different agars. So most of the time we normally use the maconche, again, because we know that Pseudomonas are non-lactose fermenters, so we'll not see we would use a maconche, but what we'd see is different colorful hues or pigmentations on the colonies that will be grown on your agar plate. So, and these colonies will have a very distinct smell, which again, for us is normally so obvious you're dealing with Pseudomonas because it's one of the only pathogen that has this distinct characteristics. So you can see that on your blood agar plate, you may see some color changes on the maconche or even on the nutrient agar. So again, you can use any of these agar plates that are available to you. So if you suspect and you've made the correct diagnosis, there are several anti-pseudomonal agents that are in use, and I've listed them here. And again, I'll send you these slides for you to take note of. And again, noting that it has a high antibiotic resistant rate. So you again look at the antibiogram of what your hospital is prescribing for use in that setting. And with that antibiogram, you're able to detect or note any of these pathogens that are antibiotic resistant. And this is a warning from the CDC again, warning that it is multi-drug resistant Pseudomonas aeruginosa circulating within the world, and the threat level of this antibiotic resistant Pseudomonas is quite high. In fact, under the CDC website, it's serious. So either we need to be able to develop a vaccine or able to prevent this infection. So ensure that the medical devices are not left in for long. If you're using contact lenses or you're using jacuzzis or anywhere that a biofilm that can be formed, make sure that the correct hygienic practices, the correct, these devices are changed frequently to avoid this biofilm formation, which then causes this serious infection that now we are seeing multi-drug resistant Pseudomonas infections circulating around the world. So with that, I come to the end of the lecture. I'm happy again to look on this chat to see if there's any questions. Again, I'll send you some notes and some literature that accompany that will take us through again these Pseudomonas infections, Salmonella infection, and also Shigella infections, how to differentiate these different areas, and I'm hoping that in this afternoon practical, you'll be able to see more of this practically. So with that, Thank you very much. I hope I didn't lose any of you, and I hope I was not muted. Okay.
No, you are not. Thank you so much.
Karibu sana. In case of any follow-up questions, you can reach me on my email here, or we can have a discussion also on Twitter, and I'll send you the notes so that you can follow through this lecture or these lectures we have been covering in the last few weeks, looking at Enterobacteriaceae. So thank you, and wishing you a good afternoon.